All right, sounds great. So yeah, thank you everybody for attending. I'm very excited to present the research that I've done in Dr. Anand's lab. So my presentation will be on the assessment of cardiomechanical function via wearable systems. And we have a lot of exciting things to talk about here. So the first thing I wanted to bring up was to motivate the research I'll be presenting today with actually a scenario that unfortunately happens around the world many times a day, every single day. In this example, we have a driver perhaps distracted by another passenger in their car or by a notification on their cell phone, taking their eye off the road and hitting the car in front of them or hitting an oncoming car. And in this example, there may be multiple casualties, and because of that, paramedics or healthcare providers may be called to the scene. So for healthcare providers called to the scene of trauma injury accidents, there's a very important step in the treatment process, which is triage. So in the process of triage, we have a lot of patients who have a varying degree of hemodynamic stability. So a patient with high hemodynamic stability might be okay to go for a while. They don't need immediate resources. But a patient who is perhaps trending towards a cardiovascular collapse might need more immediate attention with healthcare resources. So it's the role of the healthcare provider to perform triage on the scene and among multiple casualties, divert the limited healthcare resources. This is a very important step, the most essential step in treating trauma injury, but it's also among the most difficult. So hopefully nobody listening on this presentation has ever been in this situation, but the fact is that accidents and trauma injuries are just a fact of life around the world in the civilian population and a fact of day-to-day life in the military population. So according to the CDC, trauma is the leading cause of death in the United States for individuals under 45 years of age, especially in the age group up to 25 years before the prefrontal cortex is developed, obviously. And overall, trauma is the fourth leading cause of death in the United States. Globally, trauma injury is responsible for over 5 million deaths per year. A lot of these trauma injuries are due to automobile accidents in civilian populations and in military population due to ballistics injury. But the statistic that really shocked me when we were doing background literature on this was an estimated 1 in 4 trauma deaths are preventable. So the majority of the preventable trauma deaths are thought to be due to the onset of hemorrhagic shock, so due to bleeding, whereas the majority of non-preventable fatalities are due to central nervous system injuries that were irreversible. So 1 in 4 trauma deaths is quite a bit, so we want to take steps and see what we can do to prevent some of these fatalities. So like I said before, triaging trauma-induced hemorrhage is a very difficult thing to do. And a couple things that healthcare providers have at their disposal to perform triage are basic vital signs. So one of these vital signs is heart rate. But the problem is with heart rate, although it does increase as the patient is bleeding out, the problem is heart rate is a very non-specific indicator of cardiac function. The heart rate goes up and down depending on sympathetic activation and the like, not just due to cardiac output demands. So for instance, giving a thesis presentation online might cause heart rate to vary, not necessarily just trauma injury. A second vital sign that they have at their disposal is blood pressure, but blood pressure almost by definition in the context of hemorrhagic shock is a late indicator of cardiogenic shock. The reason is that as the patient is hemorrhaging, there are various compensatory mechanisms that the body employs to maintain stable blood pressure and thereby cardiac output. So the onset of hemorrhagic shock is almost by definition the decrease in blood pressure without a compensatory increase. So although heart rate and blood pressure are part of a staging system that healthcare providers use to assess the severity of hemorrhagic shock, hemorrhage, as it progresses to hemorrhagic shock, it's not ideal. So both of these indicators have problems with them. So what we'd like to do is answer the question, what if we had a digital biomarker which accurately tracked blood volume loss to aid healthcare providers? So this may help them triage patients better on the scene. But the problem here is just assessing blood volume loss wouldn't matter, and I'll illustrate why that is. So the concept here is that we have two patients who are losing blood at the same rate with each heartbeat. The thing is, patient one may be a younger, healthier individual who has a larger capability to compensate for losses in blood volume. So they reach hemorrhagic shock at a much later stage. Patient two might be an older individual or an individual with pre-existing conditions who reaches hemorrhagic shock much earlier in the process because they have less of an ability to compensate. So if we had a system that said how many milliliters or liters of blood have been lost by the patient, it wouldn't account for this inter-subject variability. So instead of asking the question, how much blood have I lost, we actually want to answer the question, how much blood do I have left to lose? This is something that's been termed blood volume status and also compensatory reserve in prior work. So in this case, we want to take the amount of blood that has been lost over the quote unquote allowable blood loss. So for this particular patient, how much blood loss would result in hemorrhagic shock, and what percent of the way are we to reaching that threshold? So the question we'd like to answer instead is, what if we had a digital biomarker that accurately tracked blood volume status during hemorrhage? And in this way, we can aid healthcare providers in triaging trauma injury. So this is really what we're looking for. The key questions that we need to answer to develop this kind of system are, number one, what are the externally measurable physiological indicators of exsanguination? So by this, I mean physiological indicators that are trackable with a wearable system. So the reason we have this question is because if we have a healthcare provider arriving to the scene, it's not very convenient to implant catheters into the patient to determine what is the true hemodynamics at play here. We need a wearable system to be able to track the physiological indicators in question. The second is, can we track blood volume status accurately using machine learning model deployed on the wearable system? So from the physiological indicators we get wearably, can we even design a model that tells us what is the blood volume status of this patient? So this is the subject of my first aim. I'm really happy to announce actually we had this work recently accepted at Science Advances that I'm about to present today. But in this aim, we like to track the progression towards hemorrhagic shock with smart wearable systems. Just a little bit of context on the background physiology. So the cardiovascular system is made up of the heart and the vasculature. And so the heart is the pump that drives the blood to the body. And there's a couple of aspects of cardiac function that we're most interested in. So one is the electrical function of the heart. So this is the timing of heartbeats that is modulated by the nervous system. The second is the mechanical function of the heart. So what is the actual muscular contraction that drives blood through the arteries and veins? The second component of the system is the vasculature. So these are the pipes that carry the blood to the body. And with the vasculature, we're very interested in the tone of the vasculature. So this could be modulating the diameter of the vasculature, or it could be due to changing mechanical properties of the vasculature, although the latter is more of a chronic process. So to develop a system that's monitoring cardiovascular physiology, we'd really like to hit all of these three components. So we're going to use three different sensing modalities to do that. So the first is the electrocardiogram, which quantifies the conduction of the electrical action potentials in the heart, which ultimately cause the muscles to contract. So this is the electrical function of the heart, modulated by either the intrinsic pacemaking capability of the heart and also by the autonomic nervous system. So for our purposes, a couple of things we like to look at is, number one, heart rate. We've already discussed some of the limitations there. The second thing is heart rate variability. And HRV, or heart rate variability, tells us a lot about the sympathetic-parasympathetic balance that's governing the autonomic function of the heart. And so in hemorrhage, we would generally expect heart rate to increase and heart rate variability to decrease as sympathetic activation is increased, although there may be a lot of confounding factors here. What we use to assess the mechanical function of the heart, so the actual heart's ability to squeeze, is the seismocardiogram. So this measures the movement of the chest wall during the cardiac cycle using a chest-mounted accelerometer. So I have an illustration on the right here. And so what this is showing is that the heart sits in a sac called the pericardium, and that's right behind the sternum. And so what we can do is place an inertial measurement unit on the sternum or elsewhere on the chest wall, actually, and using the vibrations of the chest wall, we can get some mechanical indicators of cardiac function. So I've listed a couple of those here. One is the pre-ejection period. So what is a period of time between the depolarization of the ventricles and the actual ejection of blood from the heart? So this would be the duration of time that the muscles are squeezing before blood is ejected from the aorta. And this is generally thought to be an indicator of sympathetic tone, although this is completely in the absence of confounders. The second is the left ventricular ejection time. So what is the time that the heart is actually bending, squeezing, contracting the blood to the body? This is thought to be an indicator of cardiac contractility, although, again, confounders may influence the actual relationship here. By that, I mean things like preload and afterload. So in the context of stable preload and afterload, these relationships are thought to hold true. So in prior work, pre-ejection period, or PEP, is thought to decrease, as well as the LVET. And as well, the pre-ejection period to LVET ratio is thought to increase. And I'll hit on this later, but the PEP to LVET ratio is thought to relate to ejection fraction, which is actually closely related to stroke volume, which is expected to increase during hemorrhage. So this is why we expect this relationship. There's a little bit more in the seismocardiogram because we'll spend a lot of time on this. So what does this SCG sensor actually look like in practice? I've included a couple pictures on the bottom left here. So this could be implemented as a wearable patch integrated with ECG and PPG sensors as well. In our lab, we've also developed a wearable form factor on a watch. So there's a lot of very convenient form factors that deploy an SCG signal. And on the right, I actually have an example of what an pretty much ideal SCG signal looks like on the bottom here. So we have one sinusoidal complex associated with aortic opening and a second sinusoidal complex associated with aortic closing. And with the opening and closing the aorta, if we were to get those timings relative to ventricular depolarization as referenced by the R peak of the ECG, we're actually able to calculate pre-ejection period and left ventricular ejection time and get all those mechanical indicators from the SCG. So finally, we have the photoplethysmogram, or PPG. So this actually measures the bulk change in blood volume in surface capillaries during the cardiac cycle. So a PPG sensor in our particular application is typically placed on a distal location. And when we see a modulation in the amplitude of the PPG signal, that's generally when the bolus of blood that has left the heart during contraction has reached the distal location. And so that transit time for that bolus of blood from when it leaves the heart to when it reaches the distal location is thought to relate to blood pressure. And it's actually been shown to relate strongly with blood pressure in the absence, again, of confounders. Unfortunately, we'll show also that hypovolemia is an example of a confounder in this case. But this is something called the pulse transit time. And on the right, I've shown the basic calculation of this. So we have our aortic opening as we derive from our aortic pressure waveform. And we have the R peak. So the interval between the R peak of the ECG and the aortic opening from the aortic pressure waveform is our pre-ejection period. And if we subtract that from the interval between the R peak of the ECG and the modulation of the PPG amplitude, we get the pulse transit time. So this is also an indicator we're interested in. So an overview of the process we're going to use to develop this digital biomarker. We have an animal model of hemorrhage that we used. So this is a pig model. In these pigs, I'm going to talk about this a little bit more, but we are going to drain blood at fixed levels of blood volume, so it's 7% drop in blood volume at each level. These pigs are going to be fitted with our wearable system, so the wearable multimodal system with electrical, mechanical, and vascular signals that we're collecting. From each heartbeat, we're going to collect a variety of physiological features from each signal modality, and we're going to combine those and develop a regression model that maps from our physiological features at each heartbeat to the blood volume status of the animal, which we're calculating using ground truth sensors. So to use that, we're going to use a random forest regression model, just a very highly nonlinear yet interpretable regression model for this task. So this is our overall process. In the experimental protocol, we had 6 pigs where we performed this experiment. The pigs were fitted with this wearable ECG, PPG, SCG system. At the same time, they were fitted with a catheter-based system. So we had catheters in the aorta, in the femur, and there's Swan-Ganz catheter getting right and left atrial pressure. So using a catheter system, we were able to develop reference physiological features that we were able to compare against the wearable system to really make sure the wearable system was pulling the proper physiological indicators. So that is shown on the left here in this diagram. On the right, I'm showing just some example traces from one respiratory cycle from one of the pigs. So in the experimental protocol, again, we had 6 pigs, and blood was drawn at 7% increments. So first we used an Evans blue dye procedure to get the animal's total blood volume, and then pulled blood at 7% of total volume. After 7% was pulled, we waited about 5 to 15 minutes for the cardiovascular system to level out. Once the pig had a sustained decrease in mean arterial pressure of 20%, that was termed our safety threshold. So that's where we defined cardiovascular collapse to occur. So on the left-hand side, I'm showing the mean arterial pressure trace from one of the pigs, the dotted line representing a 20% decrease in blood pressure. On the right-hand side, I just showed some examples of the physiological indicators that we pulled at each heartbeat. In the blue, we have our reference standard system. And in the blue, we have our wearable system. In the green, we have our wearable system. So we're just showing just some example traces of these features. We pulled one of these physiological features from each heartbeat. The model that we used was a random forest regression model. So just briefly to touch on it, because it'll come up later, a random forest regression model is a type of ensemble model trained using something called bootstrap aggregation. So in this case, the problem with random forest models based on decision trees is that they have a very high tendency to overfit the data. So one thing we use to combat that is bootstrap aggregation. So in this case, we take our entire training set that we have available, and we train different models on different subsamples of that training data set. So in a sense, each model is becoming an expert on a particular, perhaps overlapping subsample of the training data set. And then our final prediction is just an aggregate of our response from all of the models. So in this case, if you have a diverse enough set of models, you will adequately prevent overfitting using this method. There are a couple important hyperparameters of the random forest model. So what we did is we followed our training procedure using a leave one subject out cross-validation, and we obtained the final performance in terms of error between estimated and true blood volume status as we varied our hyperparameters. The two important hyperparameters for random forest models being the depth of each decision tree and as well as the size of the overall ensemble. And so while there weren't enough animal subjects in this case to perform a true hyperparameter optimization procedure. In this case, it didn't end up mattering too much because we hit a region of high stability in the hyperparameter space where we see that a small change in the hyperparameters themselves didn't lead to a large change in the error that we were seeing in this model. So what we ended up going with was an ensemble size of 100 and a maximum depth of 10, as it seemed to do pretty well in this just tuning step. So this was our key result from this experiment. And so what this showed us was that the externally measurable physiological features accurately tracked blood volume status using this machine learning regression model. And I just want to make a couple of comments on this from the biomarker development standpoint. So from that standpoint, what we'd like to see when we're developing a biomarker is that, forgetting about the scale of the y-axis for now, what we'd like to see is that for each intervention for which we're measuring this biomarker, that we see a statistically significant difference in that y-axis. So taking away that scale right now, what we saw is that between each level of actual blood volume status that we computed for each animal, we saw a statistically significant increase in the predicted blood volume status from our digital biomarker. So as far as digital biomarkers go, that is typically adequate. In a lot of cases with those kinds of biomarkers, there's not really a clinical correlate to the output. So it might be something like an arbitrary output or some kind of similarity score or something that doesn't really have a physical basis. So what you really want to see is just that significant difference between the intervention levels. But what we actually did see here is that our predicted blood volume status tracked the actual blood volume status very accurately in our held-out cross-validation approach. So this was an additional thing that we like to see when developing the biomarker as well. From the practicality standpoint of this, because we used a nonlinear regression model that was interpretable in terms of random forest, that it really bodes well for the clinical application of this. The reason is it's very easy to audit the regression tree model. It's very easy to see that if you made a medical error, where exactly in my model did I make that error? A lot easier to see than other types of nonlinear models that are kind of more popular nowadays. So using this type of model does have a very good clinical translation. And then third, just from a standpoint of, I think a lot of times we forget to acknowledge how cool the work we do is. So in this case, it was very interesting for me to see that we were able to assess the subject's internal blood volume status using only external sensors. So using a system that's mounted on the outside to get the internal hemodynamics of the subject, for me, is a very interesting result, aside from the scientific result. The second key important result from this was that cardiomechanical features were actually driving the performance of this digital biomarker, by far and away number one being the PEP to LVET ratio. And in fact, if we just had a system of the PEP over LVET ratio in conjunction with the traditional vital signs, you could get a very robust digital biomarker for assessing blood volume status. So this drove us to say we need to really look into more getting these cardiomechanical features in practical settings to make this research a little bit translational. So the major conclusions from this, so the wearable system achieved high performance in tracking blood volume status in this animal model. So this random forest regression model may serve as a viable digital biomarker of the subject compensatory reserve, although future research is needed in human subjects. And very importantly, among the physiological indicators studied, the PEP over LVET ratio derived from the SCG signals most strongly predicted the onset of hemorrhagic shock. So cardiomechanical indicators may supplement traditional vital signs, enabling the data-driven triage of hemorrhage. And the overall contribution of this to the scientific community, this demonstrated for the first time that cardiomechanical indicators can be measured with wearable technology to accurately track blood volume status in an animal model of hemorrhage. So this serves as a good preclinical predicate for future research. So the major question from this, we really like to see that we develop this digital biomarker in this animal model, but the question here was, what do we really need to make this technology translational? So as we saw, the cardiomechanical indicators were the most important aspect of this digital biomarker. So what we needed here were clean SCG signals, which we had, because we had an anesthetized pig lying on their back. So we were able to really get very clean cardiomechanical signals from which we derived our aortic opening and closing, and thereby PEP and LVT. The problem is that in practice, we don't really get clean SCG signals. If you have a patient who's ambulating, if you have a patient who's recently been in a trauma injury, they may not sit still for you all the time. There may be other factors at play, such as where the sensor was placed, etc. So what we need to make this technology translational is to find a way to parse the SCG signals and also determine their signal quality as well. So a couple challenges we have processing SCG signals is number one, in-band artifactual noise. So after we filter the signals, there are motion artifacts that are still in the band that corrupt the SCG signals we see. So I showed you before a very clean example of an SCG signal. This is also an example of SCG signals taken at rest. So the signals I'm showing here are separated by the ECGR peak, and I took a bunch of heartbeats, about 1,000 milliseconds each, and I just lay them on top of each other. So this is several heartbeats taken from one human subject, and the dark black signal being an ensemble average of all of these individual waveforms. So at rest, we have a very clean SCG signal. We can probably get aortic opening and closing quite simply. Then during exercise recovery, what we see is there's a little bit more complication because the signals are non-stationary. Aortic opening and closing are changing, so the ensemble average of these isn't quite as clean. And then once the subject starts getting around and moving, so in this case, the subject started squatting exercise and a walking exercise, very rapidly the signal quality of the SCG signals degrades quite heavily because of this in-band noise. We have additional challenges as well. So there's also interpatient variability in SCG morphology. So on the right here, I'm showing SCG signals from 2 different patients recorded in 5 different sensor positions. So patient 1 being in green, patient 2 being in blue. And we see that between the subjects at each sensor position, there are variations in the signal morphology that would make a simple peak counting method to determine AO and AC a little bit difficult. The second thing is that between the 5 sensor positions, especially on and off the sternum, we see high variability in the morphology of the signals as well. So typically when we're processing SCG signals, we have some combination of these 3 noise sources, among others, such as postural variability and others as well. So my first question, coming from the medical standpoint, before we even get into how do we derive cardiomechanical indicators from these signals, is how can we ensure that a biosignal has sufficient quality before we use it for life and death clinical decision making? So we can see that if we had this digital biomarker for assessing blood volume status that said treat patient 1 before you treat patient 2, if that was wrong, then that's a death sentence for patient 2. And I think that a lot of times in signal informatics, we forget what are the real world consequences of our models being wrong. And a lot of times these are wrong because the signals that we're getting don't have high quality. So in my estimation, this is something that we needed to answer before we move forward. This is the subject of my second aim. So in this aim, I developed a unified framework for both quality indexing of SCG signals and also localizing sensor position. I'm going to focus a little bit more on this on localizing sensor position, although we use this more nowadays for quality indexing because I think it illustrates these points a little bit better. So the signal quality index that we developed here is what's called an ensembled template matching approach. So I'll go through these stepwise. So with the ensembled approach, this is actually the same as our regression model before. The anatomy of an ensembled model is such that we train several separate models on different subsamples of our training data, much like we did with bagging. So in this case, with our signal quality index, our aggregated prediction from all the models was actually just an average of the individual model functions. So each individual model I'm representing here is a model function phi of I. So if I have m different models, I just take the signal quality index from each model function and then average the output. So what we really want to develop here is what is that model function that tells us what the signal quality is. So to do that, we use a template matching approach. This was chosen because it really models the gold standard for signal quality assessment, which is expert determination. So expert determination is an expert who has experience in knowing what is a known good template signal. And then based on their experience with that, given a signal X that I'm trying to assess, what is the signal quality? Does it look like the templates that I'm used to? So this is why we came up with the template matching approach. So in these approaches, we have a template signal T, and we have a signal X that we are trying to assess. So we say that the signal X, from the perspective of the template, its signal quality is a function of the inverse distance between the signals, because if the distance is higher, we want the signal quality to be lower and vice versa. So our model function phi of I is a function of that inverse distance. And so what we did with the signal quality index is we interpreted this as a negative exponential to bound the value between 0 and 1, 1 being perfect signal quality, a perfect match between the template and the signal, and 0 being infinite difference between the template and the signal. I won't get into this too.
I'm sorry about that, but in your previous slide, this template signal Ti, is that the output of model I?
Sorry, no. So the model I is parameterized by the template Ti. So we will assess the template from our training data. So that's what we gather from our training data. And then the model function is what is the inverse distance between the signal and the template.
Oh, okay.
Yeah. So the template
model basically generates this Ti and then calculates this index for that specific model, phi I.
Oh, so that's coming up. I will mention.
Oh, okay.
Yes.
Thank you.
Yeah, I will, just in a second. So to do the distance estimation, we spent a lot of time developing this, but I won't go to it too much. For pulsatile signals like cardiac signals or as well oscillatory signals, one thing we like to use to assess distance is something called dynamic time warping. So before we find a simple Euclidean or L2 norm between two signals that we'd like to assess the similarity of, one thing we'd like to do is mitigate the effect of any stretching or compressing or variation in the timing of time domain features before we make that comparison. Because as, for instance, heart rate changes, we do expect natural shifts, compression and stretching in these cardiac biosignals we're getting. So in the past, typically this kind of distance estimation was done with something called dynamic time warping. However, the problem is with SCG signals, because dynamic time warping is a greedy optimization algorithm, oftentimes features that originally correlated with each other in the SCG signal were pulled apart in order to minimize the Euclidean distance between the signals. So because of this, this wasn't an optimal distance metric to use with SCG signals, as it often underestimated the distance between them. So to start with our distance function, we developed something called dynamic time feature matching. And so this was just additional restrictions placed on the warp path that we get with dynamic time warping, so that features that originally corresponded with each other still corresponded with each other. So on the right, I have an example of a template signal in black and a signal that we got from a patient in red. And so features that originally corresponded with each other, connected with dotted lines on the top, still corresponded with each other in the bottom graph. And going back to our dynamic time warping graph, these features that were originally in alignment were brought out of alignment after performing dynamic time warping, just as an example. So with our experimental protocol, just to show the quality indexing implications of this method, there were 16 healthy subjects. So these subjects were fitted with ECG and SCG sensors, as well as impedance cardiogram to get ground truth aortic opening and closing. I won't use these here, but I'll use these later. So in this experimental protocol, there was a 5-minute rest period, followed by a walking exercise, followed by a squatting exercise, and then a 5-minute recovery period as well. So the way, to answer your question, Dr. Han, the way that we developed these template signals was to take an ensembled average SCG signal from the subject's rest period. So we used the held out validation approach here. So we had a held out subject J, where we had the SCG data from all the exercise levels, from rest, recovery, exercise. And then for each held in subject, so we had 16 total, so we had 15 held in subjects, we took an ensemble average template from the rest period. And that's just for this quality indexing, and we'll show that that might not always be an optimal way to develop a template. So we had 15 templates, so 15 model functions. And so for each SCG signal from the held out subject we'd like to assess, there were 15 SQI scores, one from each model function from each template. And so taking an average of all these SQI scores, we obtained one final SQI score for each held out subject's heartbeat. Just to show some qualitative results from this, so this is data from one of the held out subjects during the testing protocol. So here in the rows, we stratified SCG signals based on the percentile rank of the signal quality index. So we have the top 2% of the signals in the top row, middle 50% in the middle row, and the bottom 2% in the bottom row. So the columns correspond to the exercise level. To the far left, we have the resting period, and in the middle, we have recovery, and on the right, we have the exercise period. And so what we were seeing is that even in rest period, we were still seeing low quality segments. And even in the exercise period, we were seeing some high quality segments. And what we saw from the qualitative standpoint is that our signal quality index was effectively stratifying the SCG signals based on the relative quality using this held out validation method. And we also saw that the SQI scores that were generated from this method agreed with heuristics that we were getting from expert annotation of these same SCG signals. So between the rest, recovery, and exercise periods, the scores that the signal quality index was giving was matching the ground truth, which was expert annotation of the signal quality index. So we had a lot of quantitative results to go along with this as well, but just for the purposes of this presentation, I'll leave that for questioning. But for sensor localization, we just wanted to slightly modify this approach we were taking. Could we take a unified approach to signal quality indexing and localizing sensors as well? So to do that, we started with a key assumption. So our assumption was that sensor position has a consistent effect on SCG morphology. And so if you were to do a hypothesis test and try to show this assumption is true, that would be something very difficult to show. But one thing we can do is tackle this assumption by proxy. So if we develop a model that's based on that assumption, and that model performs very well for our task, that does lend support, although it does not prove the original assumption we were making. So this is just kind of the basic assumption we were starting with to develop our model. So the assumption here was that if I took SCG signals from two different locations, so here I have location green, location blue, if I had an unknown SCG signal here in black, then it should have a higher signal quality index relative to the location from which it originated. So if it was taken from position green, then the template I take from the green location should say the template in black has a higher signal quality relative to the what template blue was saying, and vice versa. So this would be saying if there were consistent effects on SCG morphology from sensor position, we could simply say take templates from different locations on the chest wall and then say which one gives the highest signal quality index from the unknown signal. That's my sensor location. In this case, to test this, we made it a little bit more complex. So there were 5 different sensor locations from which we developed templates in the same method. So we developed a single template per held-in subject for each of the 5 different sensor positions. So each held-in subject had 5 templates instead of 1. So I have this model on the left here. This is our ensemble model just for reference for what we've been using this whole time. And on the right is the model we used for sensor localization. And I just want to show the correlation between these models. So like I said before, for each held-in subjects, we had 5 templates, 1 from each of the sensor locations. So this corresponded to developing templates from a different subsample of our entirely available training data. The model function in this case, we used the quadratic discriminant analysis classifier. So that said, for each unknown SCG signal I get, I get 5 signal quality index scores per held out subject. So based on the distribution of these 5 scores, what is the most likely location to which it belonged? So in this case, we weren't simply taking an argmax. The reason is because in the real world, each template has a different propensity to give certain SQI scores. It might vary due to the quality of that template to begin with. So in this case, we trained quadratic discriminant analysis classifiers in a held-out cross-validation approach to do this. And the output of the model was just the mode. So taking the votes of these individual QDA classifiers on the ensemble, what is our ultimate prediction? So what is the prediction that was made most frequently? The experimental protocol we used to test this was much the same as the previous one. In this case, there were 10 healthy subjects fitted with ECG, SCG, and they were also fitted with ICG. And they had a rest exercise recovery protocol. So the results from this, I'm going to show here in this slide. So SQI scores can be used to infer sensor location for both stationary and non-stationary SCG signals. And I'll start with the stationary. So what I mean by stationary is during the rest period, not the exercise or recovery period. So the aortic opening and closing points are not changing. And this is much easier to do. So in this case, as I said before, we generated our templates using this synthetic approach or this ensemble averaging approach. And so this actually worked very well at rest. So in the top here, we have a confusion matrix with our true position on the X and the predicted position on the Y. And we saw is that on a heartbeat by heartbeat basis, we were actually predicting the sensor position correctly using this method quite often. And what we have on the bottom is a little bit of a different formulation of the problem. So the shading corresponds to the F1 score for detection, where the correct placement was on the mid-sternum and incorrect placement was anywhere but the mid-sternum. And we did this to compare it against prior work, which used the same data set with the same metrics. And so what we saw here was actually pretty encouraging. So as we increased the ensemble size of our model, the F1 score increased. So what that tells us is that we had an information gain. As we included more held-in subjects in our model, the actual performance of the model was increasing. So this says if we have more and more patients, we might expect to see actually a greater increase in performance. And so what we saw on the y-axis here was that because of our problem of formulation, we didn't actually have to take a beat-by-beat sampling. Because when a patient puts on the sensor in the morning, or when the healthcare responder puts on the sensor to triage trauma injury, we don't need a heartbeat by heartbeat prediction of sensor placement. We could take the first 10 heartbeats and make a single prediction on sensor placement. So as we increased the latency before we made each prediction, the F1 score for detection actually increased as well, and we saw a continuing pattern with that. The problem is during recovery, when the signals are non-stationary, we noticed that this method of taking the synthetic template was breaking down. The reason is because the generative factors of the signal were changing, and the signal was no longer stationary. Once you take an ensemble average, you're actually averaging out information from the signal, and you can no longer make a valid template. So what we did instead here is we chose templates based on how representative they were of the SCG signals as a whole. So for each SCG signal from each subject, what we did is compared it to every other SCG signal from that subject, computing the distance between that signal and all the other signals. So a signal with a minimum average distance between itself and the remaining signals for that subject was selected as an optimal template. And using this method, you can actually take more than one template per subject. You could say, return me the top 5 or the top 10 templates per subject. So what I'm showing on the bottom right here is actually the same analysis. So on the x-axis, I'm taking the number of heartbeats before I make each prediction. And what we're showing here is that as we increase the number of templates that we use from this representative method, we're actually able to boost the performance of sensor localization, not just at rest, but also during exercise recovery. What we have in the dotted line is the synthetic template that we were using on the left here. And so that underperformed even just taking one representative template from each subject. So this is to show that using ensemble template-based methods, or just template-based methods in general, the method that we use to assess distance and also the method that we use to generate our templates actually has a huge effect on the overall performance of the model. So these are two things you need to keep in mind when developing template-based methods. So the major conclusions from this: SCG signals recorded in different sensor positions were distinguishable based on their morphological characteristics. And this was the biggest kind of theoretical contribution of this work. So in this case, we not only observed that sensor position had a particular effect on SCG morphology because of the way it affected the performance of our model, but we were also able to exploit the population level patterns in the SCG morphology to do signal quality assessment and also localize sensor placement. So this was a very major theoretical contribution in this case, where we typically think of SCG signals as being somewhat stochastic and not uniformly affected by these kinds of things. And the proposed template-based method effectively stratified SCG signal segments based on the artifactual noise. I included that in here because this is how we typically use the signal quality index in all of our other SCG research, is just to remove segments that have artifactual noise. And the major scientific contribution of this, so again, we exploited population level patterns in the SCG morphology to enable sensor localization and signal quality assessment using a single unified approach. So finally, moving on to the final aim, the results in the first aim represent an optimal scenario for SCG feature extraction. So again, we had a pig lying on the back, as I said before. Instead, we need a more extensible framework for SCG processing to translate this technology. So as we saw, even with minimal movement by the patient, there are large motion artifacts in the SCG that lead to non-stationarity and also motion artifact corruption. So we really need a way to really pull our cardiomechanical features from the SCG signals in a robust way so we can enable the clinical translation of our digital biomarker. Typically, the way we do this in signal informatics is we start with our signal, and then we develop an algorithm that extracts whatever indicators we're looking for from the signal. And I've always had a problem with this kind of approach because, in effect, you're yanking the information that you want from that signal, and what we end up with is a case of mining and optimization that sometimes leads to a blind end where we can no longer proceed. So once the method stops working, it's really hard to proceed from there because we didn't glean any real insight from it. In this case, I wanted to take a little bit of a different approach when inferring cardiomechanical indicators. So what if instead of fitting the signal to the outcome, we did the reverse and we fit the outcome to the signal? So that's maybe a little bit esoteric at the moment, but I'll go into more detail on this, where we start with our signals and we assume that there is some underlying dynamics of this signal. And if there is in fact some physiological information that we'd like to glean from this signal, it should be present in the underlying dynamics of the signal. If it is not, then the signal likely does not contain that information to begin with. And if the signal does contain that information, it should be reflected in the signal's underlying dynamics. So this is the general framework that we're taking here, and it'll make a little bit more sense in a few slides. So the major assumption, as before, we're starting with an assumption with our model. So the assumption here is that SCG signals derive from simple cardiovascular dynamics. And so this is where we're coming from here. The SCG signals in the time domain are very highly complex and very difficult to process. However, the generative factors that lead to SCG signal generation are believed to be few. So in this case, we have aortic opening and closing are believed to be the major generative factors of the SCG. So in this case, we would expect the intrinsic dimensionality of the SCG signals to be low. And in fact, if we plot the SCG signals in a state space, the state space should be approximately two-dimensional and quite simple in response to single stimuli, such as a single stress event or a single exercise event. So for instance, for hemorrhage, we would expect both aortic opening and closing to decrease. So in this example of a hemodynamic state diagram I have, we'd be starting from this baseline state in the top right, and we'd be coming down to this state in the bottom left as we drew blood from the patient, because aortic opening and closing should both decrease. So this represents a very simple dynamic process, although the signals that we're actually observing in a time domain are quite complex. So in a sense, just to extend this for what it would look like on a heartbeat by heartbeat basis, so we'd start theoretically in a state as 0 somewhere in our state space diagram, and as we hemorrhage, we're coming down to a state that's further to the bottom left. So with each heartbeat, starting from our initial state, we'd have a change in state delta. And if we knew the change in state at each heartbeat, we could trace the path from our starting state to the final state. And this is really what we're trying to get at when we process SCG signals, even though we develop really crazy complex feature mining methods to do so. So this was the focus of my third aim. So how can we infer cardiomechanical function from SCG signals using these underlying dynamics? And there's two ways to do it. There's an active way where we actively model the SCG signals as deriving from these dynamics, or a passive way where we just passively observe these dynamics. Obviously, the former is more powerful, but in this presentation, I want to focus on the latter for the sake of time. So in this case, we took a manifold mapping approach to estimating specifically pre-ejection period from the SCG signal. So what that means is we started with our seismocardiogram signals, which were generated in response to some hemodynamic change, so in this case, hemorrhage. And what we were hypothesizing is that the SCG signal was actually, because of its low intrinsic dimensionality, was restricted to a low-dimensional manifold. So by estimating the distance traveled along the manifold itself, we believe that this should correlate to at least pre-ejection period and perhaps even LVET from the SCG signal. So the way we went about testing this kind of hypothesis was that we took our same pig-based hemorrhage data set from before with our 6 pigs undergoing the exsanguination protocol, except in this case, we also included a fluid resuscitation protocol. So once the pig reached hemorrhagic shock, or a predefined cutoff of 20%, we reinfused the blood that was drawn at the same 7% increments until the pig was back to the same blood volume level they started at before. And so hopefully this figure on the right will make more sense here. Sorry, on the left. So one thing I'd like to start with is if we take our signal, which is just really a vector, and we do a principal component analysis on that, and we take that signal and plot it on our PCA axes, what we see is pretty much nothing. This is really just a clump of points. But one thing that was really interesting after applying the signal quality index to this data is we saw that the signal quality index preferentially identified these points that are kind of the middle of this donut here as being a lower quality than the signals on the outside edge. And actually, it's hard to see from this diagram, but the 98% of the points actually lied on this very simple manifold in these PCA axes, whereas only 2% lied off the manifold. And those were the 2% that the signal quality index identified as low quality. So this is something that was quite curious when we started out. But what it led to was for each of the 6 pigs, we noticed that there was a very simple manifold structure for the SCG signals, although they were very complex in the time domain. And if for each of the 6 pigs we colored the SCG data points based on the pre-ejection period amplitude, we actually noticed quite a nice gradient, where as you go across the major axis of the manifold, the pre-ejection period increases. And we noticed that across all of the 6 pigs. So we were wondering, could we use the distance traveled along the major axis of the manifold to reflect our pre-ejection period instead of coming up with some crazy feature mining algorithm? And so the way that's typically done, well, there's actually a lot of ways to do manifold mapping procedures. Among them, the most inefficient and least liked is isomap, However, it is something that we used as a gold standard here because even though it takes a long time to run and very easy to break, it's very highly accurate. So I've presented that method on the top here. I won't go too far into it, but starting with our manifold, we develop a wireframe of the manifold, and essentially what we do is flatten that wireframe to come out with our graph on the right here. And so the major axis of our manifold is this variable y1 on the bottom. So what we did instead is we developed a manifold approximation method because isomap is not something that you can run on a wearable system. So instead what we did on the bottom left here is showing all of the manifolds from the 6 pigs plotted on the same PCA axes. And on the right, what we have is a manifold approximation method. So for each of those data points, we actually just plotted the point on a kind of circular basis function here and used the displacement along this unit circle to determine the distance traveled across the major axis of the manifold. So instead of having an order n cubed isomap algorithm, we actually had an order n manifold approximation algorithm. So the first one took us on the order of days to run for each pig, and the bottom one was on the order of seconds. And so what we noticed is that the change in PEP was related to the displacement along the major axis of the manifold formed by the SEG signals. Both for the isomap and the approximation method did very well in this case. So I'm showing the data from all of the 6 pigs here. On the x-axis being the latent variable that we pulled from the manifold mapping method, so y from isomap, and then we have this delta theta from our approximation method, and the y-axis being the change in pre-ejection period. And what we noticed is that actually both methods were able to assess the change in pre-ejection period using held-out validation and linear regression under a 5 millisecond error for PEP estimation. So what this was saying to us is that you don't need necessarily to take a very complex route to getting latent variables from the SCG signal. These latent variables should be present in the dynamics of the SCG signals themselves. It worked very well for the pre-ejection period, but as we'll see later, for the left ventricular ejection time, because it depended on aortic closing, a very low SNR event, this didn't always work as well. Just to revisit very briefly why we had the result we did with the signal quality index, harkening back to our model function for the signal quality index, so this theta sub I was this exponential, negative exponential of our distance. So as our distance was smaller, the model function would go to 1, and then as the distance was higher, it would go to 0. So what we did here is I just wanted to show why we got that behavior from our signal quality index. So starting with a template signal, so I'm showing this pulsatile signal on the top here, we can actually generate what's called a DTW equivalent from whatever pulsatile signal we have. So using a Monte Carlo simulation, we can actually develop a valid dynamic time warping path, and from that path generate what the comparative signal would have been. So in this case, the green signal on the left would be a DTW equivalent of the black signal on top. So if you were to compare the signal quality index of that template and that signal, it would be 1 because the distance would be 0. So for a template signal in black, I did Monte Carlo simulations here and generated a bunch of DTW equivalents for that template signal. So for this is for one of the 6 pigs in our study. And what I wanted to show here on the right, so this should look familiar. On the top, these are the SCG signals that we derived from the hypovolemia study. So in the gray and in the red, these were the SCG signals that we actually saw. On the black wireframe, this black wireframe is actually showing the DTW equivalents that we obtained from our single template signal. And what we saw is that the points that the SQI was rejecting were lying off of the manifold formed by this wireframe. So what we're noticing is that the signal quality index itself actually reflected the manifold behavior that we saw from the SCG signals just from this simple passive manifold mapping method. So the signal quality index seems to be a very natural match as we defined it for SCG signals. And on the bottom, just to briefly describe how it's working. So for a template T, we can actually look down from the top of the manifold here and see kind of a crescent shape. So if you have a signal S in green, if it lies anywhere on that manifold formed by the template, it should have a signal quality index of 1 because the distance is 0. But then if it lies off of the template, what the signal quality index does is say, first find the closest point on the manifold to that signal, and then find the distance between the manifold and the signal itself. So it's taking the template matching approach just a little bit further by incorporating the intrinsic dynamics of the SCG signal. And just very briefly, instead of passively modeling the SCG signal as deriving from this simple dynamic process, we can actually model it we can actually do this actively. And so the way we did that is using, I won't go into this, but we used a machine learning neural network-based method to do it. So we had our input signals, SEG signals X on the left, and then using an autoencoder type neural network, we had our signals X tilde on the right, which were just supposed to be the same as our input signals X. So this is an autoencoder type architecture. And what we did is for each set of SEG signals we had, we determined the initial state as 0, and then the change in state delta, so that we could fully map our state space and low dimensions. And then from our state space for each set of SEG signals we got, we actually derived the generative factors using a learned subspace for those signals. So we were explicitly mapping SEG signals as deriving from this kind of low dimensional process. And that actually enabled a lot for us, but just to focus on one thing, it showed us that SEG signals exhibit position-specific dynamics. So here on the graph on the left, these letters correspond to SEG sensors placed in the center, left, right, top, and bottom relative to the mid-sternum. And so what we saw is that the generative factors that we were getting from this model that we were explicitly mapping depended on where the sensor position was. So we would get different trajectories in the subspace based on where the sensor position was placed. And one cool thing that enabled us to do was actually adapt to the patient misplacing the sensor using an algorithmic approach. So as before, we would have to ask the patient to move the sensor if it was placed incorrectly. But using an approach like this, we can take an algorithmic approach and digitally move the sensor from off the sternum back onto the sternum to perform our analysis. And that was something that was really cool. So on the right here, I was showing just a quantitative version of that. So from our generative factor subspace, we developed a linear mapping using held-out cross-validation, where we would develop a separate linear model mapping from each sensor position to aortic closing. And so what we saw was when we matched the correct linear mapping to the sensor, the actual true sensor placement, we got a higher correlation between our estimated and actual aortic closing. So what this showed us quantitatively was that, yes, these SEG signals exhibited these position-specific dynamics depending on when they were placed. And this also worked for aortic opening as well. But explicitly modeling the initial state and the change in state also enabled us to not only see the change in PEP and LVET, but also explicitly estimate what is the actual PEP and LVET. So on the bottom left here, I'm showing what is the actual error in predicting true PEP and true LVET. And both of these are actually very good in the context of prior literature. And just to show this as an illustration on the right, so what happens when we correctly match the linear mapping from our sensor position to aortic opening and closing. So when we use the correct mapping, this is showing the points in black here. So we're actually able to map directly to our ground truth aortic opening and closing on the x-axis with our predicted aortic opening and closing on the y-axis pretty closely when we match the linear model with the true sensor position. When we mismatch the linear model, we actually end up with higher error in terms of estimating our aortic opening and closing. So these were just some very interesting results we had that we weren't able to get using the passive method of doing this. So the conclusions of this, modeling the SEG signals as samples from an underlying low-dimensional dynamic process may enable inference of PEP and LVET in a more robust adaptive manner. And manifold mapping provides a simple method of PEP approximation, harnessing the intrinsic structure of the SEG signal. So going back to what we were saying before, instead of deriving a feature extraction algorithm which yanks the features we're looking for from the signal, we're actually able to use the intrinsic behavior of SEG to do this instead. So it's a lot more simple, a lot more elegant. And finally, nonlinear dimensionality reduction via generative factor encoding actually achieved robust approximation of both PEP and LVET, and can also enable algorithmic correction for sensor placement. And the major contribution of this being that we elucidated the dynamics of cardiomechanical signals, enabling morphology and sensor position independent physiological inference. So this still needs a lot of future research, but this is showing something that has never been shown before in prior work. These are just a summary of my contributions I've already stated, so I won't go through them, but they're here for reference. And all of these aims kind of fit together in a nice way. So we'd like to develop an overall system for this trauma injury triage. We started with developing a preclinical biomarker, and so that was an essential component. And then with aims 2 and 3, we came back around and said, how do we really enable this digital biomarker, especially in the context of cardiomechanical sensing? Future directions, so we want to apply these to larger cohorts of subjects. So all the methods I described here for both healthy patients and patients suffering from cardiovascular disease in at-home and inpatient settings. So these are largely theoretical results. And as we go now, collaborations with other people in the lab, we're actually trying to translate these to more practical scenarios, and we have some pretty interesting preliminary findings. And for our blood volume status estimation, applying our proposed multimodal wearable system to assess blood volume status in human patients with progressive hypovolemia. So this will be a large study that should take place over several years. It'll be very difficult to obtain that data, but ultimately it'll save many lives. Specifically for AIM-1, one thing to note is that we used a time point by time point model instead of using a time series model. The reason is because the progression of hypovolemia in that experimental study did not reflect the natural history of exsanguination, and any transition probability you use in a state-space model will simply learn your experimental protocol. So this is something for future work as well. And methods of quantifying the subject's initial physiological state using wearable systems should be explored, because this was the major limitation of the model, where we needed to know the subject's baseline. And you might not always have the subject's baseline. So this is a major limitation for AIM-1. Just listing some manuscripts here. I'd like to acknowledge the people in my lab who helped me here, especially Jacob, who since day one, we've done everything together. So all the research that I presented here, Jacob and I worked on together, and we have a lot of projects moving forward that we'll continue to work on together. So I want to give him a special shout out. One of the smartest guys and kindest people I've known. So very privileged to work with him. My advisor, Dr. Anon, what can you say? I'd like to say one thing in particular, that he's been a role model to me as an individual and as a researcher, and it's been a real pleasure to watch him work every day. And he's really set up an environment for us to thrive. Whereas graduate students, he protects us from a lot of things and really leaves us to pursue our research interests without worrying about too many other things. So big shout out to him. Everybody else in my lab. Mo Bashir helped me get started early on. Daniel has helped me a lot along the way, and anybody else who I listed here. I've worked with you all at some point, and I'm very thankful to do so. Thank you to my committee. I worked with Dr. Roselle and Dr. Hahn very closely on some of this research, and I really appreciate their guidance. I had a lot of support from Emory as well, and I'd like to give a shout out to Chris Rofs at T3 Labs for helping us with our pig study as well. Also to my friends and family, my friends Vince, Steven, Carlos, especially those who have gone before me in the PhD program, gave me a lot of inspiration. My parents obviously have helped me my entire life. So what can I say here? In the same way that Dr. Anon set up an environment for me to do research effectively, they set up an environment for me to get my education. So this wouldn't be possible without that. And my fiance Marina has read pretty much everything I've written. So if you've ever read something I've written, she's definitely proofread it for me and given me some feedback in everything I've done so far. So big thanks to her as well. So thank you for attending my TED Talk. I will take any questions from the public at this point. Again, if you can drop questions in the chat, and then if I see your name come up in the chat, I'll open the channel for you.
Excellent.
But if not, that's okay as well.
Excellent. That's a hilarious slide right there. I was just reading. I was reading over it. That's great. Great. So let's go ahead and take questions from the general audience first, and hopefully a couple people will ask questions. It's been very difficult to get anybody to ask questions in these things.
That's fine with me.
From the general audience, but come on, guys. Does anyone have a question, please? Just make
thank
up
you to the people in chat. But Jacob. Oh,
there you go.
Well, it's bad when your collaborator is asking questions on the research he knows about.
Jacob, can't you unmute and ask that question instead of chatting?
Jacob, it's okay. So Jacob asks, which result are you most excited about? I would say for our research, the development of the digital biomarker. So the slide I showed where we're showing the actual versus predicted blood volume status was, I think, objectively the most important result from all this, because it serves as an important predicate for the clinical application of the work. Although I am equally excited about everything, but just to be a little bit pragmatic. So I was most personally excited about the algorithmic correction for sensor placement, but that's probably the most theoretical result in the whole thing. So that still needs more work.
Yeah, I guess there's. Oh, here we go. Here's another. Can you comment on the effect of anesthesia? Good question. On the preclinical AIM-1 studies versus a non-anesthetized scenario of patients presenting to either the emergency room or EMTs.
Right. So just from the most obvious standpoint, so the animal was stationary, and so that really helped us along the way. From the anesthesia standpoint, I want to point out also that the animal was on mechanical ventilation. And there were some indicators that we derived, specifically active measures of preload, that depend on mechanical ventilation of the patient, which may not always be present in that scenario anyway. So the pig being on anesthesia, being on mechanical ventilation helped a lot in that development of the model. That may not always be the case. The anesthesia did have an effect on at least one of the pigs in terms of their hemodynamics. So one of the 4 pigs, and I can show that here in a second, but one of the 4 pigs responded quite negatively to the anesthesia, where there was a large drop in blood pressure. So that pig was administered atropine at the beginning of the protocol, and because of that, we saw a very muted response to hemorrhage, especially in the early stages, because that pig was already compensating for the hemodynamic effects of that. So that affected our experimental protocol in that way. But yeah, like I said, that anesthesia affected the study in a way where we can't really expect that digital biomarker to translate for that reason, and also it was done in
What is going on?
no idea. Is that better? Okay. So it was done in an animal model. Yeah, wow. Being done in an animal model and under anesthesia, so you can't directly translate the biomarker, but it does serve as a predicate for where we want to start when we're looking in the clinical setting. So that's a very good question.
To that point, maybe this is more of a question for Jacob, actually, because it has to do with the study itself, but there was, and maybe it's more of a question for the relative hypovolemia, the choice of, I think we used, was it isoflurane, or what was what did we use for the anesthesia? I think that was an important part of that too, right?
Inhaled isoflurane for, oh wow, there were two agents. I had that here. Sorry, I can look it up if you'd like me to, but I don't have this second one on hand.
No, I think it's okay.
I wanted to show the briefly the intersubject variability I was talking about. So in this case, this was pig 4. So where I have this asterisk on pig 4. So this is showing that their initial level of blood volume loss because of the administration of atropine was delayed. So this is why we really need to know the subject's initial physiological state before we can really apply this model.
Great.
So anesthesia was induced with xylazine. Sorry, it was induced with xylazine and telazol and maintained with inhaled isoflurane.
Okay. Great. Well, I think at this point we can, I guess if anybody else has questions, jump in, but it seems like nobody else has questions from the general group. And so, thank you. Thank you. Thank you. It's been a pleasure. I wish we could meet in person right now. It'd be great to see you guys. I'm sure you're very proud. You did an awesome job. So, okay, well, I guess we'll close the session at this point to just the committee members.
Would you like me we'll stay on here and just have the committee?
Oh, yeah, stay on here, definitely, yeah.
Okay.
Yeah, stay on here.
Okay. Thank you, everybody.